up? My name is Carson Odell, formerly known as Carcesis, like everybody should know me as, and I'm starting my first episode here of MCAT review, and I'm starting off with biology. It is going to be a hard one, and if you're asking what credibility I have, it's none. I have no credibility. You should take whatever I say with a grain of salt, but even an unsalted piece of meat is still good. Gandhi said that once. Anyway, I'm going to just help you and help myself reviewing the MCAT biology books, notes, everything that I could possibly think of to help you out, and more selfishly, help me out. I'm trying to get into med school just like all of you are, and we're all going to be successful because we work together on this. Okay? Anyway, starting off, and what I'm starting off with first is, as you can see, biology. Biology to me, impossible. I hate biology. You give me numbers, you give me equations, easy peasy lemon squeezy. You give me concepts, I suck. So we're going to start off with chapter 1 of biology. And I will tell you right now, this is a long chapter. I have my little notes right here, and this isn't even all of it. This is not all of chapter 1. And it is dang double-digit pages, right? So we're going to cover part of it in this first little part, and we'll just try and grow and grow and grow, and hopefully get chapter 1 and all subsequent chapters done pretty dang fast. Anyway, if you are tuning in on this and you want a teacher that will just lecture and lecture and lecture you, just say it straight to your face, that ain't me. I'm gonna sit here, I'm gonna joke around. I was a tutor in college, and I would just joke around. There was no seriousness going on. It was just learning while laughing, and I feel that's the best way to learn. If that works out with you, epic. And let's get started. And by the way, a lot of this stuff comes from Kaplan books. I'm going to try to interweave things that are outside Kaplan in as well. And we'll see how that goes. So first thing is, the very first thing covered in the biology book is cell theory. And I think you have probably heard this said about 1,000 times, but what cell theory is basically what cells are. What defines cells? How can we define what a cell is? And you should be able to kind of start to name what you might be able to see. The first one is what? A little second to think. All living things composed of cells. And you've probably heard that 10 times in the last year. It's very common. It's the first thing you honestly think of when you think of biology, right? That, mitochondria, photosynthesis. Those are the dang three, the big three. We ain't talking anime here. Okay. All living things are composed of cells. I'm composed of cells. You're composed of cells. Plants are composed of cells. My ability to speak is not composed of cells because it's not living, but that's fine. 2, the second main theory is the cell is the basic functional unit of life. And I'm trying to write as fast as I can because I speak as fast as I can, and that makes my handwriting horrible. So good luck with that. Okay, so the basic functional unit of life, that just basically means it's the starting point, right? Everything builds off of those basic units of life. You know, cells make up everything. Of course, you know, what makes up cells, then we get into a different question, right? But cell is the basic functional unit of life. Going into the third one, we have to come from pre-existing cells. What does that mean? Pretty dang self-explanatory. We just have to come from previous cells. You know, cells come from cells. Cell replication, mitosis, meiosis, binary fission. You've probably heard of all of that before, right? So cells are coming from these pre-existing cells, and they're just multiplying, right? You can't have a cell made from a virus, right? That just doesn't make sense. Maybe you can, maybe I'm just not smart enough. But a cell has to come from a cell. And these are the three main ones that you originally started with. And then, scientists were starting to be like, I think I can think of a third one, or a fourth one, I mean. And that is going to be that cells carry genetic information in the form of DNA, and this genetic material is passed from parent to daughter cell. So going back to the third rule, we have genetic information, DNA, in our cell. This cell is then passed along to future and future generations. So DNA passed on. And I write in a weird way. I'm gonna write all my notes very simplistically. I feel my best way to learn is from the ear and just hearing things. So me writing things down, I'm gonna write very shorthanded. I'm gonna write my notes kind of just shorthanded, and a lot of the information is gonna be coming from my mouth. Some of you might not like that, I understand, but it's kind of the way that I operate. It's kind of the way I teach. So I'm just gonna try and do that way, and maybe I'll try and, implement more writing. And you will see later my absolutely amazing drawing skills. To, help out as best I can with people that learn different ways, because everybody learns in a different way. And if you are interested, I'm gonna change it to color. That is chapter 1.1. Let's go, one section of biology done. And we're moving on to the next already. And we are going to go to eukaryotic cells. So I know you guys probably already know what a eukaryotic cell is, right? But think of it here. You have two types of cells mainly, right? One is a eukaryotic, and the other we're going to learn later, and hopefully you can think of it. I'm going to give you a second before I say it. Prokaryotic, right? What is eukaryotic? Think of an example of eukaryotic before I give you one, right? Just to get your brain rolling with what eukaryotic cells are. I'm going to give you a second. And there's many examples of them, so if I don't name them, you probably thought of one that's correct, right? You have us. We're eukaryotic cells. Well, we're made of eukaryotic cells. We're not one cell. Any sort of like animal, your dog, your cat, your fish, your pet goldfish, any sort of just living thing, that is keyword going to be, multicellular, complex, I guess, more complex. But before I even dive too far into that, it looks like it's saying that I spelled wrong, but I never spelled wrong. So I'm just gonna ignore that. Okay, so main thing when we are hearing about eukaryotic cells is membrane-bound organelles. So this is just the first thing that the Kaplan book jumps into membrane-bound organelles without actually explaining what membrane-bound organelles are. So first off, organelles, another way for organs, right? Humans have organs, heart, brain, skin, liver, whatever, that's an organ. And then for a cell, they just call it organelle. So membrane-bound organelle, membrane is kind of like a skin almost. The scientific word is going to be a phospholipid bilayer, which you might have heard if you took, maybe in the your basic biology is going to biochemistry, you probably heard about phospholipid bilayer. But just membrane-bound organelles are inside the cell. That is the key differential, mainly, between eukaryotic and prokaryotic cells, okay? So let me just write that down. So our membrane-bound, we're going to have our phospholipid bilayer, okay? And our phospholipid bilayer, for any sort of eukaryotic, prokaryotic, any cell that has a phospholipid bilayer, a key factor to note is that it's highly selective. And what would you think that means, to be highly selective, right? Say I was walking into a bar or a club or anything like that, and I'm highly selective in who I want to talk to. I would just be sitting there wherever I'm at, not talking to anybody unless that perfect person's there that I want to talk to, right? I'm an introvert. I don't want to talk to nobody there, right? I'm highly, highly selective, aka not selective, right? So phospholipid bilayer is able to sort of determine what comes into and out of the cell, right? And that is a super important for sort of like maintaining homeostasis, if you remember that word from, elementary school, right? Letting things into the cell, letting things out of the cell, sort of controlling the environment is basically all in the hands of the phospholipid bilayer. If there's no phospholipid bilayer, things just go in and out willy-nilly, right? So you're not going to have as much control, right? So it's important to understand that our membrane and all of our all our cells make up our body have membrane-bound organelles, and these organelles only let certain things in and out, okay? And I'm sure we'll go into more detail about the phospholipid bilayer, especially in biochemistry, knowing like what is going in and out, what is easily able to go in and out, what is maybe a little bit more difficult. You can probably already start to brainstorm sort of things that you can, things that might be able to go in and out easier or harder, right? And next we just go into just some organelles, mainly for eukaryotic ones that are important to know. And the first one is probably the one that you're all thinking of right now, and that is the nucleus. Okay, so the nucleus contains all genetic material necessary, I don't know if I spelled necessary right, for replication, okay? And I'll zoom out so you can see what I wrote. But why did I specify necessary for replication? Because inside the cell, and I don't know if you know this or not, and we're 100% talking about it later, there's other places that hold genetic information, other organelles that hold genetic information, mainly the mitochondria. The mitochondria is a big one. It has basically its own set of genetic information inside of it. But the nucleus is necessary, and we replicate the nucleus for replication. That's why the sort of differential is there. But typically, you think of the nucleus, you think of the blueprint. It has that genetic information. It has the information for RNA. It has everything like that, okay? Another key piece of information for the nucleus is its environment. What is the nucleus, right? So the nucleus is surrounded by a membrane plus another membrane, okay? And you might be thinking, why 2 membranes? Okay? And the reason for 2 membranes is to create a nuclear envelope, okay? So I'm going to call this the nuclear envelope, and you probably have heard about that already, but it's a key piece of information to really know. We're going to have that nuclear envelope, and it's going to be surrounding the nucleus. Think of the nucleus as a ball, and inside that ball, or around that ball, is 2 layers. So I'm going to draw it, of course. I love drawing. But we're going to have this double membrane just circling around the ball. That was the worst representation I've ever done with my hands. Like, honestly, I don't know if you might have just lost brain cells from what I just did. But this nuclear envelope specifically has a separate nuclear environment. I'm just going to shorten environment to envir. Why not? So this nuclear environment, its goal is to separate DNA, and of course I'm running out of space, DNA from RNA. So hopefully you guys have a decent understanding of the difference between DNA and RNA, deoxyribose nucleic acid, and then just ribose nucleic acid. But in general, you have DNA, which is translated to RNA, which is holy smokes, that's why I'm cooked. We have DNA that's transcribed to RNA, which is translated to proteins, right? and proteins basically do everything in the cell. So we need the blueprint from the DNA to make the study guide for the protein, right? Just a short way to kind of think about it. And we'll probably beat that thing to death going over, translation and transcription. But that is for another day, okay? So inside this envelope, there's specifically going to be pores, and not the pores on your face, but pretty dang similar, right? If you were to think about these pores, right, thinking about what our membrane or envelope is supposed to do, what do you think these pores help in doing? A little drink. These pores allow for selective two-way exchange. And this is just exchange. I think of like commerce, right? You exchange something for something else. It's kind of the same thing in the nucleus. These pores are just little holes. And imagine my head being a molecule. I'm just able to go through those pores, right? So it just allows for better exchange throughout the nuclear envelope, especially with a double membrane. It can get hard to get some things through. So these pores just allow for that movement of molecules, whatever, especially ones that are harder maybe to get through that the membrane, to go through when necessary, whether it be through, protein assisted, which we'll get later, but it just helps out, okay? And of course, we are coming up on the perfect opportunity to draw. So I'm gonna go bang, bop, boop, bang. So as you can see, I just have my water bottle. That is not what you're seeing though, you're hearing that. As you can see here though, we have a very roughly made circle. This would be one membrane. This is representing one membrane. And we have another part of the circle. Bing, bong, bop. And this is up for contention with my worst drawing ever made. Here we have the nucleus, a horribly drawn one, okay? And these right here are those pores. These are the membranes. As you can probably already tell, I don't really know why I'm writing it out. The membranes that make the envelope. Perfect, okay. So we have the pores and we have the membranes that make the envelope, and this is of course going to be, and I'm making it big for a reason, don't worry about that, our DNA. Perfect, okay. So our DNA has coding regions, which are basically the regions that are being read out and creating the RNA and further proteins, called genes. And high school time, you know what genes are. They are representation of the proteins being made, your hair color, your eye color, just whatever building blocks you have of your body are made from your genes. And just other tidbits to know is that our DNA is really linear, but that linear DNA is wrapped around histones. Wrapped around histones. And then that is not it. These DNA wrapped around histones are then even further wrapped around to form chromosomes. So we have linear turn into stone wound turn into chromosome. And that's just all by, the twisting of it and the wrapping. And there's of course there's interactions involved, hydrogen bonding, everything like that's involved. But that is a lecture for another day. Okay. So we have another tidbit of information. The subsection of nucleus called the nucleolus is used to synthesize, or really, I guess, transcribe is another word to use in there instead of synthesize. That's just the book's wording. rRNA. And that is going to be about 25% of nucleus. And it is the darker bruised spot. That bruised is just a word I made up for it. Not a word I made up, a word I attributed to it. Who am I using these big words? What can I say? Okay, so that's the nucleus done. You probably heard about the nucleus 1,000 times over, and it's not a new story. Let me quickly grab a charger before my iPad dies. But going after the nucleus is something you might have even heard even more, and I'm not going to say it. I promised myself I'm not going to say it. We have, let's make it this color, the mitochondria. Okay, so the mitochondria, I'm going to speed this dang up. Nobody wants to sit here and talk to me. The mitochondria, like the nucleus, has two layers. These two layers are the outer plus inner membranes, okay? And this will be really easy to see from a drawing. So I'm honestly just gonna draw it, and then we'll talk it out afterwards, right? I'm running out of drawing space on this page. Okay. And then a little wormy guy right in here. Oh, geez. Meow, meow. Okay, so this is our little mitochondria here. Pretty dang big mitochondria if you ask me. But our outer membrane is, of course, if you think about it, it's gonna be this right here. Let me make that a different color, just so we can better see it. Let's make it blue. Actually, no, let's keep it this color. This is our outer membrane. And this is going to be the barrier between the inner part, which we're going to talk about later, and the outer environment, right? Or the cytosol, okay? Then we have in here, I'm just going to, from right there, the intermembrane space. So it's a little bit confusing, outer membrane, intermembrane space, inner membrane, but it all kind of makes sense, right? So we have the outer membrane, the outer shell of it. Then inside that, we have the intermembrane space, right? And then what's of course gonna be this little worm, or the black part of the worm, the coloring of it, that is going to be the inner membrane. So though, yes, it's just a whole bunch of words repeated, it kind of makes sense. It's not too bad, okay? And then inside the inner membrane is of course not gonna be intermembrane space. They've got to make it a little bit confusing. It's going to be the matrix, not the movie Matrix, but close enough. Okay, so those are the main parts of it. And just a region to know, and maybe not a region, but a place to also know is these little inner folds right here, where you have, almost overlapping. And of course, in an actual better drawn picture of mitochondria, you have these folds being a lot closer together. So it makes more sense to kind of point at these folds and just name this as the cristae. And something that probably hasn't come up yet, but 100% will, is I can't spell out things, and I can't say things for the life of me. I'm useless in that stuff. So you're gonna have to roll with the flow. It's gonna be hard, I promise you that. I promise you that. Okay, so the inner membrane, the main goal of the inner membrane is just to work as the barrier, right? Just separate it from the outside world, okay? The inner membrane is just gonna be the barrier. I just dang circled inner membrane when I'm in outer membrane. The outer membrane is going to be the barrier between the outside environment and the inner environment of mitochondria. The inner membrane is, of course, going to be the barrier between the inner membrane space and the matrix, okay? Something that the inner membrane does that is important to know is it has these foldings of cristae, and what these foldings do is they just increase the surface area, right? If you have one dang line like a hot dog, it's not going to have as much, surface area on the outside of the hot dog than a slinky is, okay? That's just a weird way to think about it. But the inner membrane contains molecules plus enzymes of the electron, E minus, whatever, electron transport chain, which I'm probably just going to write as ETC from here on out. Okay. But yeah, we'll cover that later. But just kind of how ATP is made. Of course, mitochondria is called So, we just create ATP, energy, make the cell run around. Okay. Another thing that I kind of hinted at before, I'm going to go ahead and make a new page for this, is, that's not the page I wanted to make. Oh, no. The matrix is, keyword here, semi-autonomous. Okay, and this means exactly what I said before. They have their own genes, and they can replicate independently through binary fission. Okay. Another kind of thing to think about, or have in the back of the mind if it will be tested or anything like that, is that the mitochondria is thought to have originated when aerobic, which we'll cover aerobic, anaerobic, everything later, but an aerobic prokaryote got engulfed by an anaerobic prokaryote. Just something to think about. And another key part of the mitochondria, which will be covered by other things, is this keyword apoptosis. And pop, not soda. You don't want to be drinking this because it's dead cells. Apoptosis is basically like the cell releasing enzymes, or I guess in this case the mitochondria, because that's what we're talking about. The mitochondria is able to do this, releases enzymes from the ETC, which ends up killing the cell. And this isn't, horrible. Of course, you think of killing the cell as being bad, but if the cell is dead weight, it's not, helping anybody at all, and it's just using resources, it's of course not going to be important to have, right? So it's important that the cell, the mitochondria, and other organelles are able to take care of and kill cells. Without that, almost every cell can be think of as a cancer cell, just being able to have full control over itself and just take energy and reproduce, right? So the mitochondria is able to combat that and, of course, in the cell. And let's go ahead and cruise to the next one. We're gonna go to the lysosomes. Lysosomes. Different color. lysosomes have hydrolytic enzymes which can break down many structures. So it's basically like the recycling bin, I guess. And it often relates to the endosomes, which we'll cover later, or really soon after this. But the lysosome, the endosome is basically like transport a lot of the material. It's like the Amazon warehouse. You probably heard of that before. It just takes different proteins or whatever, and it transports them to other parts of the cell, right? So if these sort of ran rampant and things need to be degraded, or maybe some of the products are just not good, so the Amazon warehouse sends it to the lysosome. The lysosome is able to destroy those cells or those enzymes inside the packaging units from the endoplasmic reticulum, and just kill it so it can't further damage the cell, right? You just have a whole bunch of garbage sitting around a cell. It's not going to do any good. Lysosome comes in, breaks up the enzymes, and the nutrients can be reused, right? That's why it's the recycling bin. Another thing is autolysis, aka apoptosis. And I don't think we really need to talk about this too much. Of course, something that can destroy enzymes can cause apoptosis of the cell. It's just kind of the way the cookie crumbles, I guess, right? I'm going to go ahead and stop this here. Our next bit is going to be about the endoplasmic reticulum, the Golgi apparatus, the peroxisomes, and the cytoskeleton. So just finishing up the organelles. This is probably going to be the first part of quite a few parts covering chapter 1, and then hopefully soon after that we can go into chapter 2 and so on, so on, so on. Chapter 1's a beefy one. It's a beefy chapter, so I wouldn't be surprised if this has the most parts, and other than once we get past this part, we can just kind of start to cruise through this. But like I said earlier, a lot of this is probably review. You've probably heard about a lot of this stuff before. It's probably nothing necessarily new yet. We'll get to new stuff, at least stuff that I didn't know of before studying for the MCAT, through my classes. But kind of a short review is we covered cell theory, which is all living things are composed of cells. The cell is the basic functional unit of life, and all cells have to come from pre-existing cells. Those are the main three, and then we added the third one, or the fourth one on, I keep on saying that, is that DNA is passed on throughout cells. So cells hold DNA, that DNA is then passed on to daughter cells, okay? And then we got onto the eukaryotic cell. We talked about how the differential between eukaryotic cells and prokaryotic cells is that eukaryotic cells have membrane-bound organelles. These membranes are just the phospholipid bilayer, which allow for selectivity. Things can't come in, things can't go out without the membrane having a say in it, right? Then we started going into different sort of organelles that we might have. Probably the most common one, the nucleus, containing all the genetic material necessary for replication. It's surrounded by the nuclear envelope, which is just two membranes. It has pores inside of it, which further allow for selective exchange in and out of the cell. It holds DNA. DNA is, of course, deoxyribonucleic acid. And DNA has coding regions called genes. These genes just basically make up whatever we have in the world. Like whatever makes us is all from these genes. And then just going into the sort of nitty-gritty with DNA, we have linear DNA wrapped around histones, further wrapped around each other to form chromosomes, right? And that's where you get the chromosomes, what you've heard of before. Then we went into the mitochondria, basically mainly the structure of it. The mitochondria has two layers, the outer and inner layer. And from this picture, we can kind of see which one's which. We have the outer layer. I'm gonna go ahead and use this right here, just the outer part separating the mitochondria from the outside environment. the cytosol. We're separating from the intermembrane space from the cytosol. That's kind of the definition of the outer membrane. Then we have the inner membrane, which does the same thing, but it just separates the matrix, which is the super innards, from that intermembrane space, right? Inside the mitochondria, we have these cristae, which are just folds. These folds allow for further interaction with the intermembrane space, and we're mainly talking about the electron transport chain. I don't think we covered it much in this first chapter, but a basic understanding of it is we have just the movement of electrons across that intermembrane or inner membrane allows for ATP to be generated. That ATP is, of course, used for absolutely everything in the cell, so it's super important to have, okay? Another thing to have about the mitochondria is that its its genes replicates independently through binary fission, which we're, of course, going to talk about later, mainly in the bacteria section. But, yes, it has genes that it is able to replicate independently, and the mitochondria can importantly cause apoptosis, aka killing the cell, right? Then we got into the lysosome. The lysosome has hydrolytic enzymes. These can break down structures, and it's super important that it goes kind of hand in hand with a lot of other organelles in the body or in the cell. So these hydrolytic enzymes break down structures that are released mainly by the endoplasmic reticulum, right? So the endoplasmic reticulum puts different enzymes in these vesicles. These vesicles then go around, different organelles in the cell. The lysosome attaches it, sends these enzymes out. These enzymes destroy whatever enzymes are inside those vesicles or whatever is inside those vesicles. And then we can kind of recycle whatever we have. If it's not necessary, it can go to other cells, basically just nutrients for other places around the cell. Think of the cell kind of as a city. Everything has its role in the city, and of course we're going to have to review that at the end of this sort of eukaryotic chapter, because it's important to know how the organelles in the body and in the cell, holy smokes, in the cell interact with each other. And mainly we're getting into sort of the main part of that with the endoplasmic reticulum, which we're going to cover next. The endoplasmic reticulum is kind of my favorite organelle. I just like it. It just be cool like that, But overall, that's sort of what we reviewed today. A little bit longer than what I was hoping for the amount of material we covered. So in the next episode, I'll try to go a little bit faster, just so we can kind of start chugging along through this stuff. I don't want it to be, dragging along, and, we all got things to do, right? And of course, this is my first, episode, so if you have any, advice, tips, things that you might want changed to help you facilitate your learning, let me know. Like I said, I've tutored before, but that's all person to person, one-on-one, one-on group. Doing it, with nobody in front of me is a little bit different, so I can't, judge how things are working, with you in the moment. So I want to make sure things are going well throughout this all. If you think I talk too much, if you think I write too much, let me know. And of course, I want this to be a facilitated learning area for everybody. I want, if you have certain techniques or strategies or advice, help others around, and let's overall make it positive, make it enjoyable for everybody. And I'm sure that over the next few weeks, we'll be able to cover biology, move on to chemistry, physics, and yeah, everything that is available for us. I hope everybody has a great day. You are all amazing people. You all have bright futures ahead of you.